what I really enjoy, the part of CMC I really enjoy, of course, CMC is chemistry, manufacturing, and controls, and we like to think of it as the stuff. You know, this is the stuff that we give the animals and we eventually give people, hopefully, when a drug gets approved. And so, you know, the drug substance, of course, is the active ingredient, and then the dosage form is also called the drug product. And so I think, you know, early on, I really love supporting projects early on. Right now in my consulting, I've got a couple of companies that are, you know, at the stage where they've got hits, they've got leads, and they're trying to select compounds. And, you know, well, this compound's got, you know, the best activity against the target, but it's got really poor, you know, properties in terms of formulation. And so how do you manage that? And when do you know to go to a backup compound? And obviously there's no easy answers. And it's not like you can apply a particular formula or process to every situation, but there are things that you can apply probably universally. And so, you know, one of the things that we get involved in, you know, early on with CMC, of course, is that when somebody identifies a hit and a compound that they're thinking they want to maybe take to an IND, you know, nobody does anything unless we have compounds. So immediately, even before you start, you're behind the 8 ball. And everybody is looking to you because you're the rate limiting step. And it can be pretty tough because a lot of times the milligram quantities that people can use to, you know, verify that they've got a hit, and, you know, do some very small, you know, in vitro type assays, even some in vitro metabolism. The milligram quantities that can be used for that, you know, are often produced by a chemical route that might have a lot of difficult synthesis steps, poor yielding, and then sometimes they have to use column chromatography to purify these intermediates, and suddenly, you know, you have needs for grams or even kilos of material, and you have chemistry that really can't produce that. And so it's a pretty tough challenge right from the start. One of the things that you know, often happens too when you're sort of in compound selection stage is that, and I referred to this a little bit earlier, is that sometimes the compounds that seem to be the most active against the target, and maybe, you know, successful in a cell-based assay, are really, you know, people use the term, are not really druggable. And so there's a lot of things that you can do to sort of work through that. But I think one of the things, and this has come up on some projects with MCI, is one of the things is that you have to really decide on your route of administration early on, and you have to kind of be ruthless with that decision. If you really need an oral molecule for your indication, you know, then you really need to look at, you know, as early as possible, you know, oral bioavailability, you know, in a mouse or a rat, and then maybe even a dog, which will take a lot more material, obviously. But if that's what you've got to have, then, you know, it can be very helpful to generate data, you know, with an IV or an IP administration. It can be important to do that to show that your compound is actually getting to the target and is actually, you know, exhibiting the pharmaceutical action that you're looking for. that's very important, and don't want to ever discourage people from doing that kind of work. But once you do that, then, if you really need a different route of administration to move the project along, you need to work through that and make that one happen. So yeah, it's a really difficult time, but an exciting time, when you're doing CMC work, because everybody's looking to you. They've all got their hand out, and you're really the rate limiting step. And companies that are, public companies, or not necessarily public, but are, private companies that have boards, and they've thrown an IND date out there that we have to meet, that can be pretty stressful. So, once you get through that and you start doing formulation development, that's another, area. And I'm working on a project right now, which probably will be an issue for a lot of your development, is that, the amount of material needed for IND enabling tox studies is usually pretty large, and it's oftentimes a challenge to make that much compound. And as soon as that compound is made, people want to start the tox study. And the issue with that, of course, is that, especially like for something like IV or IP administration, the vehicle that you use for toxicology studies has to really represent the vehicle that you're planning to use, in people, especially if that vehicle, contains some excipients, that might be less than tolerable. Because, FDA wants to see that the animals got similar exposures, not just the TK analyses, but in terms of excipients that humans are going to receive. Now, if you're developing an oral dosage form, you get a break there, because pretty well accepted that if you're doing that oral formulation, or your product's going to be dosed orally, you can dose your tox studies with simple solutions or simple suspensions, and you can do just fine that way. And then if you develop a tablet or a capsule that has different excipients than you use in tox studies, nobody's too worried about that, as long as your excipients are, typical sort of excipients used in solid dosage forms. So with oral, you get a bit of a break there. Now, if you're doing something very unique with your oral formulation, then FDA is going to expect that, you provided the animals with that same sort of unique presentation then that you're going to give people. And so the idea there is pretty obvious, in that, FDA wants to make sure that the animals have been exposed to the drug, at high levels, and that, when we start into humans, it'll be at a safe dose. Just as an example of that, I filed an IND one time, and we had micronized the API before we gave it to the animals, and they wanted to make sure. Well, we were micronizing the API before we gave it to humans, and they wanted to make sure that we had micronized the API that we delivered in tox studies. We, of course, had. But there was just an example there, because they were fearful that humans might, naturally get a lot bigger exposures than the animals. So, but anyway, I'll stop there. I mean, that's just kind of a very quick overview of life in the CMC world, especially supporting early stage projects. But it's the area that I like working in the most, because I think it's the most creative and the most challenging. So So anyway, I'd love to hear about some of the things that you are running into or have run into, or any questions you might have.
I was following all your talk, and one of the main questions when we are talking about contraceptive targets is the way of administration, obviously, but it's not only the activity, like if they go through the testes or not. The blood testis barrier is a problem, and then you have the blood epididymis part. A lot of times, this is one of the questions that review panels ask, is like, how do you do for the drug to the testes? So,
I'm sorry, I'm having a hard time hearing you.
sorry, it's about the delivery within the testes, passing the blood testis barrier, because for male contraception, that is a very important point. What are your thoughts on that?
Yeah. So I think that's that's obviously very important for that project, and that might be a very good example as to it might be very helpful to deliver some drug IV or IP or sub-Q, to deliver that drug in a manner which you're getting you're getting blood levels, and you're giving the drug an opportunity to cross the blood testes barrier. But probably, and this might be true for a lot of the projects, for male contraception, it's probably going to need to be an oral formulation to be successful. That may not be 100% true, but yeah, I think that makes perfect sense. But once I knew what blood levels I needed to get through to cross the blood testes barrier, and then hopefully to see the actual pharmacological action, that you're looking for, then, that gives you a good benchmark as to then what you need to achieve, with the dosage form that you're actually looking to bring into phase 1 studies.
Can I perhaps just build on that question? I was wondering, if we have human PK data and we have in vitro against our target IC50 data, what is the multiplier that you typically need for human PK versus in vitro IC50?
Well, I've never been able to figure that out, but it is a problem. It is a typical problem. And what I think most people use, IC50 data, to do the med chem work, and to look at, just, rough inhibition, comparative inhibition among compounds to the target. But I think once you do that, then you really have to just go to the animals. Rats probably first, mice or rats, and then maybe other animals. But you really have to just go to the animals, because there'll be a little bit more, then you can do allometric scaling, to get to humans. But I've never been able to think about the leap from IC50s all the way to human PK.
Can I also just follow up on something that you brought up about doing animal studies, and try and get your opinion on more perhaps, which was, if you are to do an IND enabling studies in animals, what material is required? So must it be GMP, or is it sufficient for it to just be research grade?
Oh, it's not required to be GMP for sure. Some people, especially people maybe that have more of a big pharma background, will tell you that it needs to be. And one reason I think people say that is because what's very concerning to FDA, and I think, the more the less serious the disease, the more this is true. And of course, male contraception is way on the end of the scale of, you definitely don't want to do anything harmful in dosing. So you have a high bar there in terms of safety of your compound. And as such, FDA is really going to be concerned about the impurities that are in your material that you used for your IND enabling tox studies. And they're going to want to know that your humans, the first in human trial is not going to expose humans to any impurity that the animals have not been exposed to. And so that's the important part, is to characterize that tox material so that you can prove that to FDA that the GMP material that you're going to dose humans, in terms of its impurity profile is a subset of what the animals have received. And in fact, I almost think that when you send an IND to FDA, they have such a little time to review it. They only have their 30 days. I want to think the CMC reviewer at FDA goes right to that data in the batch analysis table and looks at that. And if that looks okay compared to the GMP batch, they're not going to, it's very unlikely they're going to have any kind of problem with anything else that you have in your application on the CMC side. So I think that's the tricky thing, is to make sure that you've got that covered, because I've seen INDs rejected on that basis. And actually, that's one of the only bases I've seen INDs rejected, is not having that safety coverage that FDA is looking for.
That was the section I was going to ask you to talk about a bit too, Lex, because I know that was helpful for us to know that, especially from a cost perspective, it's not necessarily required that you start out with GLP studies, that you can do some initial analysis and make some steps before you do transition into the GLP stage of studies to characterize and make sure that your lead is a good bet before you start doing the more costly GLP studies.
Right. I mean, the typical thing that people do, and I've talked with Heather and Logan a lot about this for some of the projects, and I think one of the really important things to try to do is to, sit down with all of your scientists, from different disciplines, on your project. Chemistry people, biology people, and try to work through for your compound, I don't know, to call it a flow scheme or a flow chart, if you will, but sort of decide, this is the gamut of testing that we're going to run our compounds through, and to make sure that we've covered all the bases before we sort of decide to take a compound to an IND. And so, a lot of times you start out with the IC50 type assays, and you develop say a top 5 compounds from that, cell-based assays. And then if you've got, and I'll use oncology as an example, if you can do a xenograph study, I think everybody knows what those are, right? Yeah, if you can do a xenograph study in a mouse to show activity against a human tumor type, say, that might be, an early indication. Before you do that, you might do, some IV versus oral, if it's an oral drug that you're looking for, PK in a mouse. Very early on, you're going to want to do some safety pharmacology like a HERG, non-GLP tests, again, very quick, not very expensive, and an Ames assay to see if your compound's mutagenic. and you're going to sort of work through your compounds this way. Most people will do one of those Eurofins 300, target panels to look at off-target effects of your compound. Almost nobody that I've experienced ever sort of, shelves the compound because of those type of assays, like the Eurofins type target, looking at different targets, that off-target toxicities. But those can give you an indication of maybe things that you might see in tox. If you see an off-target toxicity that could relate to blood pressure, and sure enough, in your tox species you see blood pressure, then that's the time to be concerned. People do CYP450, inhibition, and again, all this is good information, but not critical. The HERG and the immunogenicity assays, those are pretty important, to get out of the way and hopefully show that you're non-mutagenic and that you have a very low HERG signal. And so it's going to be different for different compounds, but I think the process is probably not going to be different. And I think it's important to sort of try to put that down on paper and to try to be religious about using it. You can change the model and decide, this is really not where we need to do this. We need to do this later. We need to do it earlier. But I've seen a lot of times where, people don't have that sort of, regimen to sort of go through, and they let some data sort of lead them sort of down a wrong path, and they make decisions based on the data, but decisions they make is what testing to do next, but then they sometimes, again, they might get too far down and then realize that they've missed something that's important. Oftentimes, the last thing that companies do before they say that they're going to file an IND is they'll do some sort of short-term tox study, usually in a rat. Again, it'll be like a non-GLP study. But what they're looking for there is, anything that really stands out. Like a lot of times what they're looking for is liver enzyme elevation and things like that you're not going to see, with a single dose. And so, once they've gone through their battery of compounds and, battery of testing, non-mutagenic, they don't have a HERG signal, then they do the 7-day or 3-day rat or something like that. Then they'll launch into the IND enabling studies. Now, that being said, most people will do dose ranging studies, non-GLP, before they do their GLP, say rat and dog studies. And the reason for that, of course, is that they want to get a sense for their maximum tolerated dose in both the rats and dogs, if we're going to use those as the typical tox species, because they want to make sure that in their IND studies, they actually see toxicity. And I've seen this mistake made many times. INDs get rejected because people didn't dose their drug high enough to show toxicity. And, the perfect tox study is not the animals all do well. The perfect one is the top dose animals, don't do well. Some die maybe, or have serious problems. The mid dose, animals, they show some effects, but they're still okay. And of course, the low dose animals possibly show no adverse effects. That would be a perfect study. You obviously usually don't get that. That's really what you're trying to achieve with those studies. And so the other thing, and I'm living this right now with one of my projects, is the chemistry is really difficult. It's like 12 steps. We've got some chromatography steps. We're going to have a hard time making large amounts of this material. And so people are trying to say, well, how much tox material do we need? How much do we need? You know, well, the answer is, well, it depends on what your doses are. And of course, your dog studies always drive the amount of material you'll need because of how much dogs weigh compared to rats. And so, you know, if you think, well, I'm thinking 300 mg per kg might be my top dog dose, and you don't get toxicity at that level in your non-GLP studies, and you got to go up to 1,000 mg per kg, which happens more than you think. Obviously, now you need 3 times the material, and you don't have 3 times the material, and you're scrambling. So those are all the sort of issues that you run into, you know, when you're sort of in this, you know, late development. Not late development, but just prior to IND studies.
So I might actually just use this opportunity to just ask about this a bit further, because, and perhaps this doesn't relate directly to male contraception, but we are in a position where we're formulating a drug that's very insoluble into a solubilized form, and we're actually hitting maximum solubility, and we're still not seeing any toxicity. So what would you recommend we do in that position? Is your idea that we still try and increase dose until we see some sort of toxicity in order for an IND enabling study, or in order to see some toxicity before moving on into GLP study?
Yeah, I mean, that situation does happen, and you know, I should preface this by saying, obviously, I'm not a toxicologist. So what I'm relaying to you is just the experiences that I've had being on these project teams and being faced with certain issues. But I think, you know, I think there are obviously dose limitations depending on your route of administration. You know, generally speaking, several routes of administration, you really can't dose more than 10 mLs per kilo. And, you know, if you've got maximum solubility in a vehicle and you've got maximum amount of dose volume, and that's all you can show, and you're still not getting that toxicity, I think there are ways forward on that. And a lot of the way that you can get through that is by is by having good TK analysis of your tox animals, so that, you know, you can show increased exposures, and then, you know, hopefully you might have to actually, you know, FDA may buy all that. If I was going to go in with an IND when I didn't have signs of toxicity, you know, that's when I would definitely set up a pre-IND meeting and have that discussion with FDA, because, and recommend a course of action. And I think there is a course of action, and FDA might come back and say, well, you know, you didn't show toxicities. You did everything you could to do that. You didn't do it. Well, we may start out at less than 1/10 of the top dose in humans, and we may ask you to generate, you know, PK data in humans in the first human study, in the first human subject rather, you know, and as a way of sort of, you know, making sure that the animals were exposed to much higher levels than your initial human subjects. So, and that is an interesting point, you know, about pre-IND meetings is you really only get one of those meetings. And there's 2 times when people generally do those meetings. One is a time like this, when you're getting ready to do your IND enabling tox studies, but you have issues, you have concerns, and you're doing something that may not be standard. Then you definitely want to get buy-in before you spend, you know, $500,000 or more on IND enabling tox studies. The other time people have pre-IND meetings is to flesh out their clinical program. So they have their tox studies done, or they're underway. They may have some preliminary data, but they're not worried about the acceptability of their tox studies, but they want to make sure that, you know, FDA is okay with their clinical plan, their first clinical study that they're going to execute based on those tox studies. So you have to make a decision, you know, whether you want to go in before IND enabling tox or after.
It's just putting you on the spot, Lex. Do you know, like, roughly what folks can, and this is helpful for us as well. Oh, actually, my first question is, can you explain what HERG is? I don't actually know what HERG is.
Oh yeah, it's an acronym for human eragogo something. It's a real weird acronym, but yeah, it's just an in vitro test that looks at the effect of drug concentration on the sodium ion channels that you know could affect and produce a QT interval issue. So the thing people are looking at is you know, they want to have if they have a HERG signal, you know, they want to make sure that it's at a concentration that is way higher than their drug is ever going to experience, you know, in practice. Or ideally, there's just no HERG signal at all. And there's been a lot of effort that people have put into trying to figure out which functional groups on a molecule or what aspects of a molecule can predict, you know, HERG activity. And people haven't been very successful at doing that. So that's why, you know, if you're in a particular scaffold, if you're looking at a particular scaffold of compounds, and you test HERG in a couple of these compounds and they show no HERG signal, you know, then you might say, well, the scaffold's not going to produce a HERG signal, and that may or may not actually be true. So I think that's something that a lot of people try to do early on to get a feel for that, because it's a definite liability. It's not a liability you can't get over, but if you've got that positive HERG signal at a low concentration, then FDA is going to make you, you know, even back up. The next study you obviously do a lot of times prior to an IND is a cardiovascular telemetry study in dogs, where you increase your drug concentration, you know, up to a single dose MTD, and you try and you look at, you know, effects on QT of the dog, and you know, you can do that test and it can be 100% clean, but if you had a positive HERG signal, you're still going to end up having to do some monitoring of humans in your first IND study, first in human study. So yeah, it's a pretty important safety pharmacology assay to check the box. Yeah, we don't have a problem there, hopefully, and then move on.
I actually know HERG stands for, it's an old Drosophila mutant name called etheragogo.
Yeah.
When they etherized Drosophila, they would lay on their back and kick their legs like they were dancing. So they named it etheragogo. But it's an important heart check. It's an important cardiac channel, actually. So that's why it's tox. But so my question actually for Lex is, if you have a small molecule that checks all the boxes, that's, you know, it's your in vitro tox studies all look good. How many, and it's just a small molecule in the CMC process leading up to the pre-IND toxicity, what percentage of molecules in your experience actually hit a hiccup and fail because of scale up or GMP manufacturing and things like that? What's sort of the how much of a bottleneck is there at the step you're focused on?
Yeah, I mean, I would have to say it's very rare to see something just totally go belly up because of the CMC issue. I totally agree. And most of the, like one of the things I always mentioned was, if you have a lot of purification steps that require column chromatography, a lot of times, and it's almost universally true, is that with enough time and money, you can solve these problems,
Mhm.
and get CMC, taken care of. It's just sometimes, you don't plan on having to spend as much time and as much money,
That's fair.
to solve these things. But you're right, very few times are there really a problem. I wanted to mention a couple quick things that I didn't want to forget to mention. And a lot of people, when they're doing this flow scheme or this flow chart I was talking about, there are several studies that people do, in vitro studies, ADME in vitro studies. And it's not that there's anything wrong with these studies. It's just that I think sometimes people put more weight in some of the results to these studies than they should. And the ones I'm talking about are like CACO2, permeability, microsomal stability, where you incubate your compound in, say, rat, liver, rat, dog, human microsomes. Protein binding is another one. Plasma stability is another one. And it's not that you shouldn't do the studies necessarily, and it's not that you should ignore the results, but almost all of those studies, say for an oral drug, all of those facets of those in vitro ADME studies, they can all sort of be combined together if you just make a little bit more drug and do a PK study in an animal. I've seen several times where, somebody would do liver microsomes stability, and you'd have this, it seems like the compound gets chewed up, in minutes. And oh my gosh, we can't develop this compound because, as soon as it's dosed and absorbed, it's going to be metabolized and it's going to vanish. And I would just say, that may or may not be true. Liver microsomes are not exactly an animal, and there's so complicated that the things that can affect PK and that kind of thing, that I would just recommend people trying to make enough drug to do a mouse or a dog, a mouse or a rat, PK study. This last company that I worked at before I retired from there and became a consultant, the guy that was head of medicinal chemistry there, he almost he got to the point where he didn't even do those tests because he was much more concerned about the PK properties in a living animal. And I think he'd just been seen so many times that these tests weren't very predictive of what actually happens. So again, it doesn't mean that you don't do them, but I definitely wouldn't shelve a molecule, for a result that was not optimal in some of those tests. A good example of that, of course, is protein binding. There's several molecules on the market with very, that are approved, very good activity, that are, 99% protein bound. And there's reasons for that, but those molecules wouldn't have been developed had somebody decided, not to develop anything that was, say, higher than 95% protein bound. So, that's just my opinion on those types of studies.
Lex, this is Logan. I had a follow-up question for you kind of on that, as well as thinking about, I think that was Lonnie's question. Sorry, I'm on the phone because I'm managing a toddler. How do you think a male contraceptive changes, kind of the previously established knowledge on what is, considered a go, no-go decision, or how much weight you put on certain assays? Do you think that the potential for limited safety tolerability by regulatory puts a specific hurdle around male contraception? And if so, what do you think that means for these sort of considerations?
Yeah, I mean, I would think they have to be. I think they would have to be extremely clean, in terms of safety and tolerability. Because contraception is, you're trying to prevent something, but you're not actually treating a disease. The more serious your disease is, the more latitude, FDA will give you on tolerability and toxicity. And so, yeah, I think that's a high bar in terms of safety that male contraception is going to, experience when you go through that, when you go through the development process. But of course, you don't really know about human safety until you do the human studies. And so, but yeah, I do expect that will be, that will be a hurdle for sure.
So then kind of to follow up that up then with a philosophical question, do you think that the first male contraceptive to actually get into human studies might be a sacrificial lamb of sorts, something that allows us to actually understand what that pathway does look like, and it may or may not be a successful pathway?
I'm not sure, it's possible, it's possible that the first one could, there could be some tolerability there. I mean, again, all toxicities and intolerabilities aren't created equal. If we have a male contraceptive that, was efficacious and tended in a certain percentage of people to increase triglycerides or something like that, or blood pressure or something easily monitorable, I think those types of things, could be, I think those types of things could be tolerated and may be okay. But then, maybe this is where you're going. The second or third generation male contraceptive drug would then have to be better. So the bar would continue to get a little bit higher.
Awesome, thank you.
So I'm noting that we have about 10 minutes left. So I just wanted to make sure there aren't any lingering questions that people want to make sure to get answered.
I was going to mention one quick thing that I forgot to mention earlier. And that is about oral drugs, because a lot of these programs may end up with an oral drug for this indication. It's really hard to discount, drugs early on because of poor water solubility, or if the log P is too high, you say to yourself, well, this drug will never get absorbed because it will never get dissolved. And, that is very possible. But I can just tell you in my experience, and this personal experience with development compounds, some of them get absorbed, and I can't tell you why it happens. Very lipophilic. When you do a dissolution test, you have to come up with all kinds of things to add to the test just to get your drug to dissolve so you can test it, and I think I mentioned this the other day with Logan and Heather was, the drug Lamisil is high log P, completely insoluble. And yet it's a solid dosage form for toenail fungus. And it's been around a long time. And had somebody thought that was not developable because of its poor solubility, it obviously wouldn't have been on the market for the 15 years or so that it has been. So So oral drugs, you know, you have to actually, you actually have to do some studies and try to and look at the absorption, because sometimes some drugs will really fool you. The good thing is, if somehow they do get dissolved, you know, the higher the log P, generally the more permeable they are. So a lot of times, if they do get dissolved, they will be absorbed. But, you know, oftentimes you might think that drugs will not be soluble enough to be absorbed, but sometimes they fool you and they actually are.